on biotechnological applications in Ayurveda and biomedical sciences, coupled with the annual meet of Society for Biotechnologists India. As we navigate an era defined by rapid technological advancement, biotechnology plays a pivotal role in shaping our future. This session will shed light on some groundbreaking developments in this space. To ensure the success of this session, we are privileged to have an outstanding chairperson and co-chairperson today with us. Our chairperson for the session is Professor Dr. Dinesh Yadav, sir. And we have our co-chair, Professor Dr. Ramwant Gupta, sir. And also we have Dr. Ankita Mishra, ma'am, faculty, biotechnologist, as the rapporteur for the session. So kindly I request all the chair and co-chair and madam to please proceed on the dais. Professor Dr. Dinesh Sharma, sir. Professor Dr. Dinesh Yadav. Sir is a distinguished professor of biotechnology at Deen Dayal Upadhyay, Gorakhpur University. Sir serves as a director of the Research and Development Cell, Dean of the Faculty of Rural Sciences, and Nodal Officer of the IPR Cell. Specializing in molecular biology, plant biotechnology, and enzyme technology, Sir has published over more than 100 research papers, 50 plus book chapters, and 3 edited books to his credit. Professor Yadav has delivered more than 100 invited lectures at various national and international platforms. Sir's contribution to higher education and research have earned him prestigious awards, including the Bharat Shiksha Ratna Award in 2012, Dr. P.K. Gupta Vikshit Krishi Vigyanik Prasad in 2015. Sir also received the recognition for excellence in training, teaching, and research. Professor Yadav continues to mentor students and lead impactful research in biotechnology and intellectual property rights. So we have Sir as a co-chair for technical session first. Along with it, we have Dr. Ramwant Gupta, sir, as the co-chair for the session. Dr. Ramwant Gupta is an associate professor of botany at Deen Dayal Upadhyay, Gorakhpur University. Sir is also the coordinator of the Institute of Agriculture and Natural Sciences and director of the International Cell. Sir has previously held the faculty position at the University of Guyana and Fiji International University. Additionally, Sir has been associated with IARI New Delhi and ARO Israel, holding a specialization in plants physiology, genomics, and bioinformatics, with reference to sustainable agriculture and plant adaptation strategies. Sir has more than 100 research papers, several book chapters to his credit. Recognized for his contribution, Sir has received the Research Fellow Award in 2024 to 25 and the Young Scientist Award in 25, and recently, very recently, just a week back, Sir recently got the Human Excellence Golden Award by the United States of America. And now we invite Sir and thank you. It's a pleasure to have you, sir, on for the first technical session. Along with it, we have Dr. Ankita Mishra, ma'am, as a reporter for the session. This session will feature insightful lectures from eminent scientists and research in the field of biotechnology, biochemistry, and molecular biology. We are privileged to have with us today leading experts who will share their groundbreaking research. I look forward to an engaging and enriching discussion. Thank you, sir. We have our first lecture in the series by Dr. Chanchai Boonla, Faculty of Medicine, Department of Biochemistry, Chulalongkorn University, Bangkok, Thailand. And today, sir is going to present his lecture on the topic HydroZitLa, Phenolic profile and medical benefits for inhibiting calcium oxalate stone formation, decelerating aging process and extending lifespan. Please join me in welcoming Dr. Boonla.
Good afternoon. It's a great honor today to get invited to come. Oh, do I have to? Can I start it now? Yes. Right. Okay.
Yes.
Sorry, I'm a little bit nervous. Yeah, I would love to say thanks to Professor Idadios, the long-term friendships. We first met in multiple universities many years ago, and yeah, we still keep in touch, and we are good friends now. This is the first time coming here, and very excited, and even more thrilled after I have to deliver the lecture after Professor Lakhotia, as he gave a very excellent talk this morning. So I'd like to start with the question. Do you think what do this celebrity have in common? Does anyone want to make a guess? Huh? Yes, they are suffering from kidney stone disease, urinary stone disease. So that means anyone can get the stone disease, right? I'm sure some of you here will get some stone disease sometimes. I don't know. Maybe I will get sometime too. So urinary stone disease is a stone happen in the urinary tract like this, and so what you're gonna use when you get a stone disease? What you're gonna take? A traditional medicines or the modern medicines? What do you think? So we have talked about this thing, both patients and doctors, and ask them what you're going to take. Yes, of course, if you talk to the doctor, they want to use or prescribe the modern medicines. But for the patient, they think different way. They like or they prefer to use the traditional medicine much more, because one thing is about, of course, when the doctor prescribes modern medicines, they have to use it. When they get feeling that it's not effective enough for them, or some patients have side effects. So that's why they look for or they seek traditional medicine. So what we think is, what if we can combine them together, make traditional medicine and put modern medicine inside? Maybe we can have a link between the doctor and the patient, make a bridge between these two sides and make exception or acceptance. So that's what I want to share to you today is about the idea that we have to make HydroZitLa. We want to combine traditional medicine and modern medicine together. And I'd like to go a little bit deeper about kidney stone disease. It's actually the ancient disease. Stone disease, the first stone in the human history discovered in Egyptian mummy. That means 7,000 years ago, we already have this disease, and it's still with us today. Why we can't eradicate it? Why we can't stop it happen to mankind? So that one is our goal. We want to stop stone disease happen to mankind, but it's not that easy. When you get a stone, the main symptom of stone disease is very painful. It's unbelievable pain that you can get, as something like we asked a lot of patients, women patients, they said it's much more painful than giving a childbirth. Is that it's undescribable pain. And what can help that? Yeah, if you have stone in the kidney, the only way that you can get rid of it out is to do a surgery. So the doctor has to do the open surgery, or some it's the surgery like that to take the stone out. And another problem happens. The stone it cannot be completely cured, so it can come back again or the recur again. So half of the patient will come back to the hospital to treat another stone in few years later after they get the first stone off. And it's not happened only in one country, one population, it's the world population problems. So in Thailand, we have population about 70 million. So the prevalence of stone disease is about 17%. That means 17 people in 100 people will get stone disease. So the population, we will get 12 million people, stone patients in Thailand. What about the world population? So it about, if you calculate it in 7,000 million people, so about more than 700 people will get the stone disease. What about India? Yesterday I looked into the data of the preparation of stone disease in India. It's about 12%. So the Indian population is very huge, right? So that means almost 200 million people in India will get stone disease. That's a huge problem. And we did a lot of research on stone. We want to know how it actually happens in humans, what the mechanism underlying the stone disease. So we did analyze a lot of stone, and then we found that calcium oxalate is the major component of kidney stones. And it's actually happened by calcium in the urine combined with oxalate. And then they get crystal. They cannot solubilize and they're treated by urine. So they get crystal and then injure the kidney cell, and at the end it will form the nasus and become a stone sometimes. And in our urine, we have a very good guy here we call citrate. This can be inhibitors. So when calcium and oxalate come to bind, if you have enough citrate in the middle, so they block the binding of calcium and oxalate. So citrate is a very good compound in the urine that can inhibit stone formation. Because calcium citrate cannot be crystal. They solubilize in water and then they get excreted. And the other thing that, so to get this crystallization, there's two components here, the concentration of calcium, oxalate, and water. So if you don't drink enough water, your urine will become more concentrated, and then it gets crystallization. And the other thing is low citrate intake, or the concentration of citrate in the urine is too low. So you have less inhibitors in the urine. And the third one is about oxidative stress. Because when the crystal here happen and then can injure the cell and then get oxidative stress, because we measure a lot of marker in the patient, and we found the patient have very high oxidative stress radicals and really low antioxidants. And what the solution now, that the modern medicine, they prescribe the medication called potassium citrate drugs. So they supply citrate inhibitors here, but we think maybe it's not enough to correct only one risk factors. So that's why we come up with the idea how to correct these three factors at the same time. So we want to make the patient to drink more water, to have enough citrate, and we need to have antioxidants in the regimen as well. So what is the problem of the current technologies or solution now? Because this medication, the patient have to take at least 3 to 6 months to get to be able to prevent stone formations. But the bad thing happens is some patients have side effects, and the test of medication is not enjoyable. We cannot imagine if you want to take this medication for 3 to 6 months. That means every single day you have to take this medication. It's getting boring, and it is getting fed at some time. At the end, the problem is here. The patient cannot comply to the doctor prescriptions. Or if you don't want to take a medication, you have to change your lifestyle. You have to drink more water, eat more citric acid or citrus fruit, or change completely the lifestyle to be able to prevent stone disease. It is the cheapest way to do, but it's very hard. So we talk a lot to a lot of patients. They can change the lifestyle within 1 or 2 weeks, and after that, the lifestyle come back normal again. They drink less water again, eat the food that can cause stone disease again. So our idea is we want to find alternatives or something new that, like I told you before, we want to combine traditional medicine and modern medicine together. And the modern medicine is molecule that is citric acid. And we think what traditional medicine that we want to use and combine with that modern medicines. So I look into the literature. The biggest traditional medicine in this world is from China and India, right? They have a very good record of Chinese medicine and also Indian traditional medicines or Ayurveda. And I come up with a very nice, I love this paper. It's published in 1994, long ago. And what they use is they use banana stem juice. It's a community recipe to prevent the stone disease. And the data is quite nice. They did it in the animal model, and they said it can prevent or inhibit stone formation in the rat. So the reason that I interested in this banana stem because we eat a lot of banana in Thailand too. We have banana farm, a lot of, and it's our daily life as well to eat banana. Like here too, yesterday I came from the airport, yeah, and I see a lot of banana shop on the way. So our lifestyle is not that different. And the other reason that I choose banana because I don't believe in rest. I believe in if there's something useless to someone, it's always useful to someone else, something like that. So for example, like we eat banana today, and after that we get peel and then we throw it in the garbage bin, and the fly and the microorganism, bacteria, then they're happy. Oh, this is my food, right? So it's become gold for them. And after that, they digest it and then it becomes fertilizer. And we give it to the plant. And the plant grow and give fruit to us again. So that's not, actually, I don't believe in rest. Rest can turn to something else as well. Like in our case, banana. There's a lot of farm in Thailand, but after they collect or harvest the fruit, and the banana tree become rest. Right? They have to cut it down and sell it at a very cheap price for animal feed, or sometimes they just throw it away. So we want to change here because the thing that we want is here, banana stem. We want to turn banana stem rest into something that have medical value. Or we can, after that, then we can have something that can help them to prevent stone disease. So that's the story, and then we come to our solutions. So we made HydroZitLa based on that criteria. We have traditional medicine and modern medicine, and combine. We think with this a perfect combination between traditional and modern medicines. And we filed patents, and then we did some shows. And what about efficacy? Yeah, I did some. We have to test it, like Professor Lacotia in the morning, that we have to prove and show them this is effective. So we did a preclinical study. My student did animal models here. They feed them to get the stone disease, and then after that, they treated with HydroZitLa and the drug that the doctor used in the clinics. So what we see here, I don't want to go too deep in the detail. What I show you here, this is a normal control. And if you see here, the white stuff here, this is the crystal, calcium oxalate crystal. So this group of animals get stone disease. Yeah, the black one here, this is stone. And after the animal get HydroZitLa or the drugs here, there's no crystal in the kidney of the rat, of the animals. So that means our regimen or our HydroZitLa can block calcium oxalate formation or kidney stone formation by calcium oxalate. And what about inflammation in the kidney? So this is the stone animal. They have a lot of inflammation here, and after they get HydroZitLa or the drug, there's no inflammation. The good thing about HydroZitLa is can reduce oxidative stress, but the drug cannot. This the thing that we think our solution is better than the drug that is used in the clinics. And then we move to the clinical phase 1 in healthy volunteer to test whether our HydroZitLa have toxics to the humans or not. We have this data is not published yet, but we're about to. So we show that HydroZitLa there's no toxic to the human. Of course, this number one, no side effects. And it can reduce oxidative stress in the plasma. And also, the good thing is here, urine volume. We have the data that show that HydroZitLa, which is due to the component in banana stem, they have a diuretic effect. They make you pee more, and then it can excrete the crystal in the urine. And we also test the capacity of the urine that can calcify calcium oxalate is significantly reduced after the healthy volunteer get HydroZitLa. The urine citrate also increased compared to the placebo. And because the HydroZitLa is combined with the herbal extract from banana stem and something else, so it contains a lot of antioxidants. So we also are whether they can have a function to delay the aging process or even to extend the lifespan of animal. So we did it in C. elegans here. This is compared between the control, the worm without HydroZitLa, and we put the HydroZitLa into the worm here. Look at here. The lifespan of the worm extended about 30%. That means if your lifespan is 100 years, you can live 130 years by HydroZitLa. This is the animal model. So that's what it means. And if you look at here, this is control. The green thing here is the aging pigment. If you're getting age, you have the green pigment accumulated in the body. I mean, this worm. But after the worm gets HydroZitLa, the pigments has become less. Or that suggests that HydroZitLa can delay the aging process. We also have the cell model to show that the cell aging or cell senescence can be inhibited by HydroZitLa as well. Also here, the data here, we measure the telomere length because when the cell become aged, the telomere will be shorter and shorter. And then when the cell gets straight and then put HydroZitLa in, it delay the shortening of the telomere. That what we think this could be delayed. And we have some data on animals. So we feed the animal with the sugar that make them age. So this is the aged animal. And this is the control. And this, after the aging animal get HydroZitLa, is become look. But I have to tell you, this is a very preliminary study. You can't take that as granted. But we can see some, it could be a beginning of the story that we can dig down into what happened inside the animal, that why HydroZitLa can also delay the aging process. So one thing that we think is about phenolic compounds in the herbal extracts, because we all know that phenolic compounds in plants, they have a property of antioxidant. They can inhibit oxidative stress, and at the end, they can delay the aging process. So that's why we have the objective to identify what phenolic compounds in our HydroZitLa. So actually, the main ingredient in HydroZitLa is banana stem, but we also have the blue pea flower extract and also some plant extracts here inside as well. So we did HPTLC here to see what is it, the component, the phenolic compound component is here. You see a lot of things here, but I want to make the long story short. So we see only about 10 compound or 10 band of phenolic compound in HydroZitLa that derive from these 3 plants here. And what are they? We want to know. So and we have the standard here. This is a routine. At least we know this one, the routine here, and this one routine, and the other one that's basilins. This is from sap plant, and this is from banana stem, and also from butterfly pea. And we want to profile it, whether what components in here. So we use this data show that the routine is actually inside the HydroZitLa. Yes, we use MSMS. And then we move to top MS analysis to see the overall or the total compounds in the HydroZitLa. So we profile phenolic compounds here in HydroZitLa and also the main ingredients, banana stem, butterfly pea, and also the subplant root. So I want to conclude it. I don't want to go to the detail too much. Yeah, this is the main compound that we see in our products. The first one is procyanin B. This would be come from the butterfly pea and basilins, epicatecins, camphols, and quercetin. Oh, I forgot lutein, the other ones. So now what we are going to do, we're finding the way to to elucidate what is the mechanism of HydroZitLa that can inhibit cell aging or aging process in animals. So this is the thing that we are doing in the lab. So we're trying to identify the mechanism, because the only way that we can convince the doctor to use this traditional medicine is to provide evidence. So we have to put a lot of energy to provide the data. It has to be a very, very validated data. So I'd like to end my talk. So the message that you can take home today is about HydroZitLa that we make in the lab, and it's become commercials in Thailand now. Yeah, it's actually a Ayurveda-based regimen, because the ideas, the concept, is come from Indian traditional medicines. And we try to prove, at least in our preclinical models, that it can prevent stone formation in the animals, and also they have the function to delay the aging process or extend the lifespan in animal models as well. So I'd like to thank all granting agency and my team, my students, and the patient, the participants in the projects, my colleagues, Chulalongkorn University, where I work. And last but not least, I would love to invite all of you to go to Bangkok to visit my university. This is Chulalongkorn University is located in the downtown of Bangkok. So it's very crowded and traffic jam with this area. But you will see this scene in the night is quite beautiful as well. So if anyone want to come, so just, yeah. Oh, down there, right? Okay. You can come to me and then text me if you want to join. Yeah. Thank you very much.
Thank you, sir. Any question for sir? Yeah. Please bring the mic.
You made hybrid with natural and synthetic compounds. How you created this hybrids?
It's very simple. We just mix things together. So we have an extract from natural compounds, but from banana stem is water extract actually, but we have our own process how to make the extracts. And then
my question is, as there are several active compounds in a concoction of a natural herbal product. Basically, it is a herb that is consisting of several kind of active compounds, and you have selected the potent one, and then you made a hybrid. I think this was the project. You made hybrid with your synthetic compound. So my question is, how you selected that compound, and then how you made the hybrid?
we didn't select that. We'll use the mixtures, like Professor Otias told in the morning. The mixture would be much more. We can't just pick the very active. I don't know what's the active compound here, but it's a mixture one. At least I know phenolic compounds that may have antioxidant to explain the effects that we see in animals. But actually, which one is actually active compound that inhibit that process? We don't know actually, but we know it's a mixture of extracts.
Why I asked you this because what we do mostly, we select a lot of databases and from there we take out either the natural products and the already commercially available medicines also, and then we create hybrids of them and then we select them. So basically at that time you will get lot of data about your hybrid molecule also, that how they are binding with your natural and the synthetic part, because it is basically a concoction. You don't know the actual active component.
Yes, yeah.
Thank you, sir.
Yeah, I'm sorry. I'm not a bioinformatics, so I don't know the docking process much. But yeah, if we know what compounds, yes, we can make the hybrid, or what you call hybrid, right? But I call myself, it's a mixture of things that deliver the effects. Thank you.
I just
So
have
we
one
have a-
last question. Yeah, so very nice presentation. I just
Thank
wanted
you.
to ask you, like, did you try in your preclinical models to reverse the stone? Does it only preventive measures, or
Good question.
if you can reverse the established stone, it dissolve it or whatever?
It's actually the prevention model. It's not cure or treatment model yet. So, but the good thing is after we launched the products, there's a lot of patients calling to us. Oh, I get the stuff. The problem is exactly the same, because the doctor prescribed the medication and then they cannot tolerate it. They have side effects. They look or they seek alternatives. So that's why they call us. And then they provide them. We provide them we give it for free. Actually, we want to know what they affect. And some patient with a small stone in the kidney, and they take this for months. And after that, they go to the doctors because they schedule for surgery. And then they go back to the doctor and they do X-ray again or CT scan again. There's no stone anymore. So that the evidence, the clinical case, give us that at some point, at some size, small stone size, it can dissolute or it can make it smaller, and then it get blood out of the system, urinary system. So, but for animal, we don't have the treatment model to explain that. But in clinical, we have, yes.
Yeah.
Yeah, we have time limitation. You can discuss later on. Please, remaining 3 other presenters are there, so you can discuss later. Thank you.
Excuse me, sir.
Yes.
I want to ask one question.
Later you can discuss.
Okay. Thank you, sir. Thank you, sir, for your wonderful lecture shedding lights on the benefits of HydroZitLa contribute to cellular protection, slow down organ degeneration to mitigate debilitating diseases. Now we have with us Dr. Sumadee de Silva for the second lecture in the series. Ma'am is a senior lecturer at the Institute of Biochemistry, Molecular Biology and Biotechnology at the University of Colombo, Sri Lanka. We are honored to have with us Dr. Sumadee de Silva, a distinguished researcher from University of Colombo, Sri Lanka. Her expertise lies in molecular biology and its application in personalized medicine. The topic for her presentation today is the science behind personalized medicine. Let us welcome Dr. de Silva.
So, good afternoon, everyone. So, thank you for the introduction. So, first of all, I would like to thank the organizing committee. It was really nice to see the decors and the effort you have taken to make it success. So, at the same time, I would like to thank you all for inviting me as a speaker.
pointer said, and it will go forward, it will go back.
Yeah, this is the highlighter. Yes. So today I'm not going to talk about anything about my research. I'm taking a kind of a general concept, what is called personalized medicine. I feel most of you like it, right? So I'm basically talk about the science behind the personalized medicine. As a molecular biologist, I would like to put forward what sort of a science we can aid towards the personalized medicine. So before going further, I would like to kind of give an introduction because she has given an introduction about me, but I'm from Colombo, Sri Lanka. So my university is University of Colombo. So you can see some of the photograph of the University of Colombo, where I'm under the University of Colombo. It's called Institute of Biochemistry, Molecular Biology and Biotechnology, that is abbreviated as IBMBB. So it is a postgraduate institute where we are conducting several MSc programs, plus we are doing MPhil PhDs, as well as we have recognized by the Queen Elizabeth Commonwealth Scholarship. So annually we are getting a Commonwealth student for our MSc studies. So this is the content, though it is like a kind of a lengthy content. I'm going to talk briefly about everything in order to make it towards the personalized medicine. So first of all, we will take the concept personalized medicine. So earlier it called as precise medicine. Now they have changed it to so-called personalized medicine. So what do you mean by personalized medicine? So it, as name implies, it's called personalized. That means it's customized. So if you look at it, personalized medicine is a tailor-made medical approach. The point is not working. So as I mentioned, the personalized medicine is a customized version, so-called tailor-made medical approach for the disease diagnosis, prevention, and treatment based on what? Because we know the medicine we are nowadays following, we will take the disease and we are try to find the solution. But what do you mean by personalized? Then we have to go for the individual. So individuals means we have to look at the individual genetic profile, molecular characteristics, the environment around that person, as well as the lifestyle factor. So you can see in this diagram the current medicine where we call as one treatment fits all. That means we consider not as an individual, we'll take the disease and then we'll find the therapy or the treatments. So if we take the treatments for the disease, you can see some of them nicely responsive for that disease, while some of them not having anything, no responsive at all. But there's another kind of an adverse set where they're showing adverse effect on those therapies. If that is the case, what we can do in future, that is what I'm going to answer today, that is the personalized medicine. There, we are not taking as disease, we are taking the individual as the subject. Then based on that, we are doing the individual profiling, and based on that, we try to find it out, the correct, the accurate medicine towards that to make them all effective for the treatment. Then in order to go for the personalized medicine, then we have to see what sort of input, what sort of things we should look at them. So as I mentioned, the individuals with the individuals, we have mentioned we have to look at genetic profile, we have to look at their lifestyle, and we have to look at their how they will be the other factors of them. So those are called omics data. So if you look at the topic is role of multi-omics data. So first of all, we will try to understand what do you mean by omics. So omic is field of study involving large-scale data analysis of biological molecule. Then it's coming the large scale of data. It is only genomics. No, it should be the contribution of all the omics data. Here I mentioned genomics, transcriptomics, proteomics, metabolomics, epigenomics, microbeings like that. Several omics data together, we have to call them as multi-omics data. So if we wanted to go for personalized medicine, we can't depend on one omics, but we have to consider all the types of omics data together. Then after that, what we can do? Then if you take Justin, it is a huge data embedded within one omics. Because of that, then in order to get one picture, we have to integrate them. So here in this diagram, is integration, everything will support in for the personalized medicine. The next question is how we are going to integrate them, since there are a large set of data inside. For that, we are using so-called bioinformatics. I'm not a bioinformatician, so I'm knowing what's behind this as a molecular biologist only. So bioinformatics is a kind of very important kind of a field which is capable of integrating the multi-omics data by using bioinformatics algorithm, like pipelines like that. So they are having different algorithms which I am not good in. So they are giving a kind of a comprehensive idea about the whole biological system. So here I have mentioned that bioinformatics can capable of contributing for the genomic sequencing, pharmacogenomics, biomarker discovery, AI machine learning, as well as what I said very early, it's the integration of multi-omics data. So you can see I have given the source as well. So this is the way they are saying how to integrate them using the bioinformatics algorithm. Then we'll go for the next slide, talking about the biomarkers. If I go back again, you can see one of the outcomes through what we are getting through by the bioinformatics is the disease biomarker identification. So before going into the role of biomarkers, then we have to understand how the bioinformatics will be helpful. Just now I mentioned it in the previous slide as well. First, we have to collect the data from the multi-omics sources. Then we have to do the processing, and we have to do the integration via using the artificial intelligence as well as machine learning sources. Then we have to go for the biomarker discovery and validation that should be done. Thereby, we try to understand the disease-related genes, proteins, and metabolites. Then those biomarkers which are identified should be used for the personalized medicine. So you can see in this simple diagram, it will be explain everything. You have to consider the patient-wise. Then we have to collect the samples, whatever the biological samples available, plus other factors, non-genetic factors and environmental factors. Then we have to do the molecule analysis, then data analysis, and then we have to go for the biomarker identification. Based on that, we can go for personalized medicine, but this is not that much easy as we are telling. Then I here put forward some of the biomarkers using for the different purposes. So first one is about the diagnosis and disease prediction. This is with respect to the early detection using genetic biomarkers, such as BRCA1 and 2 for the breast cancer. Then treatment selection using targeted therapies based on the biomarkers and the drug responses using the pharmacogenetic biomarkers. So example I have given here is the HER2 positive breast cancer patients responds well for the Herceptin. So likewise, I have jotted down several of the factors where you can use the biomarkers, such as monitoring treatment effectiveness, reducing adverse drug reaction, because you know when we are treating with drugs, thinking that this is very much effective, but it will show some sort of adverse, I think I have mentioned earlier also, because of some sort of a genetic biomarkers, we have to check because some genetic variants make those drugs adverse. Then actually these are few of them, there are more of them. So the last one I have mentioned is the guiding the lifestyle and preventive strategies, because some of the biomarkers, if you check, you can see some of these genetic variants will tell you this particular, because of this particular variant, you will get the disease. Based on that, we have to change our lifestyle and what are the food we should take, what the way we have to follow likewise. Then when I summarize all the biomarkers, what I have mentioned here, we can broadly categorize like this. This is a nice diagram, though it is very simple. Some of the biomarkers we can use as a prognostic. What do you mean by that? Is it likely to develop this cancer? That is a question normally we are having. Then diagnostic purposes, we can use the biomarkers. What type of cancer is it? That is the diagnosis purpose if you are using the biomarker. If you using the biomarker for the prediction, it says, is this the optimal drug for my cancer? Then some of the biomarkers we can use as a pharmacodynamics. So what is the optimal dose for my body? And here, I have mentioned lastly is the biomarkers we are using for the recurrence to check it whether it says will the cancer returns. Then next will be, now we understood that we should need omics data. We have to integrate them via the bioinformatics algorithms. Then we understood through that we can able to identify the biomarkers, and those biomarkers will be helpful towards the shifting towards the personalized medicine from the current medicine. Then next one is how we are going to get this omics, multi-omics data. So for that, we are using so-called high throughput technologies. So what I'm going to talk here is some of the example about the high throughput technologies. So first of all, we try to understand what do you mean by high throughput. So it refers as the ability to process large volume of data. That's true. We understood that several omics data containing lots of data. So by that, we have to use those technologies to handle larger volume of data. So by using these technologies, we can make it the personalized medicine enable rapid large-scale analysis of genetic, molecular, and cellular data. So here you can see some of the example of the high throughput technologies such as NGS. Everybody's knowing, I think India are very famous for that. Microarrays, mass spectroscopy, high throughput sequencing, single cell technologies. That means cell-wise we can do the analysis rather than taking as a tissue or blood. We can go by cell-wise and do the analysis. So here this diagram says the very simple steps. This is not that much easy, which I have seen shown in this diagram. Simple steps where we are towards the next generation sequencing workflow. So since I have talked about the next generation, I will move forward with some of the next generation platform we are talking about. We are talking about the genome sequencing. We call whole genome sequencing, exome sequencing, targeted gene panels. I know this audience is very much knowing about this next generation sequencing method. But the thing is, what we should know is using these each and everything, it's based on our purpose and as well as our pocket. So if you take the whole genome, you know whole genome means coding, non-coding, all of them. Not only that, nuclear genome plus mitochondrial genome, we have to sequence. We can get the data. But if you look at the depth-wise, it is not comparative to exome sequence. It is much more lesser. But cost-wise, it's very expensive. And if you look at the exome sequence, if you know a place or we know a coding region where we need to get a kind of idea about genetic, we can go for the whole exome sequencing. Depth-wise, it's much more bigger than the genome sequencing. Then targeted gene panels are the ones which are known as panel studies. Normally, you know that most of the companies, they have constructed the panels for that. That means they have either taken the gene or part of the gene which is responsible for that particular disease. So those are called targeted gene panels. So based on the purpose of us, we can select which next generation sequencing we are using for. Then next topic, I'm going to talk about the pharmacogenomics. Now we know about the omics data integration, then doing the bioinformatic analysis and everything. What going towards is the identify the correct drug. So pharmacogenomics, if you say, it's a study on how the individual genetic makeup affect their response to the drug. That means based on whatever things, though the same drug, if we like apply for anything, it won't be effective because that I have shown you in the first slides as well. So everything depending on the gene behavior. So you can see it will be play a kind of a key role of helping the tailor-made drug treatment for each patient, which is improving the efficacy and reducing the effective adverse effects. So if you take this diagram, you can see this is the individual. So based on that, we are doing the omics data analysis, and you can see the same drug according to this diagram. So, which you can see in different individuals, the way we are giving is different. So, then this is one of the example I have given here, is the variation in CYP450, that enzyme family will be matters for the effect of drug metabolism. So if you are having an idea about these things, this is one of the examples I'm providing. So based on that, we can judge what sort of a drug or what sort of a drug we should use on the drug response on that particular individual. Then I'm going to, whatever things we so far get in, we try to understand what sort of applications we can use this personalized medicine. So first one, I'm talking about the oncology. That means in the cancer treatments and prevention. So you can see one of the example I have given here is the BRCA1 and 2. I can say just one. BRCA1 and 2 mutated cancers responds very well for the PARP inhibitors. So that type of a thing, if we are knowing the status of the BRCA, then we know what sort of a drug we should use. Otherwise, if we commonly use it, it won't be effective. So the second one is a pharmacogenomics. It is again what I said just now discussed is a personalized drug response. So one of the example it has been provided. Then I'll go to the third one as a title wise. So infectious diseases. We can use the we use the personalized medicine in India, like it's precision treatment and vaccine development. Then fourth one is cardiovascular diseases, that is risk prediction and therapy personalization. We can do it. Then the fifth one is neurological and physiotherapy disorders, that is tailored treatments. Sixth one is sorry, this point is not very. Sure. Yeah, fine. Yeah, it's then and there it's working. Right. Then and there it's working. It's then and there working. Yeah, without that I was right. Point is working. Yeah. Yeah, that's fine. Okay, I'll do it. Fine. Yeah, sure. Okay, right. Thank you so much. We are going back, sorry. Then we will be here. Then I want to talk about the sixth one, where the application of personalized medicine is the autoimmune and inflammatory diseases. Seventh, rare genetic disorders, where we can use the personalized medicine, early detection and gene therapy. Eighth, reproductive medicine and fertility treatment. 9, metabolic and life-based medicine, we can so-called nutrigenomics, where we can make the application as the personalized medicine. So I'm not going for the details of these things. So, but one thing I want to talk about is this. So in this, we can say, as I mentioned here, the personalized medicine consider the individual, not the disease. So based on the same thing I'm emphasizing again and again, we have to go for individual wise, not as a disease. Then whatever the concept, the new concept comes, along with that, what is coming is the challenges. So now I'm like, this is kind of my latter part of my presentation about the challenges in the personalized medicine. So if you take the first one is the limited access to the genetic testing. We know genetic testing is nowadays is kind of a trend. The clinicians are much more learned towards the genetic studies, and they are knowing what is genetic testing means. They are like to look at the genetic testing reports and everything. Though that much of exposure is there, the patients are sometimes unable to afford for those. So that is one of the challenges in towards the personalized medicine, because this is a concept we are still not established fully. The second one is data privacy and the security, because we know it is an individual data we are dealing with. If that is the case, keeping them stored is some sort of a problem for the privacy as well as security-wise. The third one is the complexity of the genetic information. That means if you try to understand the genetics, sometimes those are very complicated because some of the genetic factors are linked to some specific health outcomes, because it can't be very direct ones. Sometimes it's very along that we cannot explain sometimes. Then the fourth one is a high cost of the personalized medicine. So you can see some of these personalized medicines are very expensive. So it is not very much common among people because they should afford it. Then the fifth one is a limited clinical guidelines because still it is kind of emerging field. We don't have a proper guidelines to control them about the genetic informations and everything. Sixth one, the ethical and legal issues. Because if we are doing something, we should have a proper ethical and legal coverage where we can see these personalized medicine still having these ethical and legal issues. Then the seventh one, everywhere in all population, we can see the cultural and religious sensitivity. Some of these cultures, some of the religions, they are not allowed for these kind of genetic testing, as well as that if it is stopped, it is kind of a barrier for the personalized medicine. Then finally, the future direction. So this is, though I said future direction, still some of these places they are doing the CRISPR-Cas activities. So then the advancement of CRISPR-Cas gene editing, it's keep on advancing. So it will be something curative treatments for the genetic diseases. And other future directions we are knowing is the artificial intelligence or machine learning. That means we will kind of training the machine in order to identify the risk. For that, we needed a large amount of data set. So that is some sort of a thing. That means without a clinician, sorry if the clinicians in the audience, sometimes without a clinician, we can train the machine to identify them. Then the expansion of the pharmacogenomics. So it will be expanding and that will be allowed for the personalized drug prescription. Then the fourth one, expanded focus on preventive medicine. So these omics data and everything will shift in towards the preventive healthcare rather than reactive treatment. I hope you know what is the difference between preventive and reactive, because normally nowadays what we are following is reactive treatments, where once you get the disease only, we will find in the solution. But I think as the previous speaker also mentioned, we should go for the preventive ones. That is the future trend. And lastly, the integration of multidisciplinary approach. We need, as a genetician or the molecular biologist, I can't do everything. There should be some sort of a strong group where we having a contribution with geneticians, bioinformaticians, and pharmacological clinics. Everything should be integrated for this. With that, I would like to say thank you.
Thank you, ma'am. Thank you for your enriching words of wisdom and on personalized medicine. Any questions for ma'am? Okay.
Yes, yeah, that's true. Yeah, it's a target group is students, yes.
Thank you so much, ma'am, for your wonderful presentation.
Okay.
I wanted to ask, what kind of work do you do in personalized medicine?
Yes, since I'm a molecular biologist, I should not say genetician, it's having a broader meaning. So I'm doing genetic studies. Normally I'm focusing on cancer research as well as rare genetic disorders in children. So actually, in our studies, if it is MPhil PhD studies, basically we are going to look at the genetic biomarkers for the early identification in Sri Lankan population. Because you know, if you take some sort of a marker which is highlighted in other population, we can't take it as ours because genetic pool is different from different. So in our cases, we are going to change, looking at the Sri Lankan population. For the Sri Lankan population, we are looking for the genetic markers for the early detection specifically. Yes.
Any markers that you might have found or you've come across in your study in cancer?
I would say we have done a kind of a comprehensive study with respect to P53. I know P53 studies are very much popular everywhere, but they haven't done very well a study in Sri Lanka, checking for the so-called hotspots regions. But it's true, hotspots region in P53 is already identified. It's exon 4 to 8 or 5 to 7. So we wanted to check our population, how it's happening in among 4 cancer types. We also highlighted the fact that they are in the hotspot. So likewise, we have done several other research and looking forward to some more.
Thank you.
Okay.
I just have one question because most of the time the students they always have some sort of like personalized medicine. It's a very good topic.
Yeah.
But what is the status at present? For example, if you're talking in India itself, whether we are following this personalized medicine or it is just in the initial stage.
Sorry, sir. Is it like if I ask the question again?
I just want to know what is the status of the personalized medicine.
Okay.
Because whether we are following this or it is just in the picture that we are just doing research in that part,
Yeah.
but it has not come to the commercialized portion, and most of the people they are not knowing about it.
Yeah, true. Yeah, true. That's true. The personalized medicine still people are not knowing because the physician also still stick to the normal the one treatment fits for all. So actually, we should make the awareness first of all. But making the awareness among general public, I don't think so because they can't understand. Other thing is, if we try to establish that thing, they may not be affordable. First of all, we have to make it affordable for the people. And for that, we have to find it out like kind of a very affordable genetic markers, first of all, for that particular given population, because it's population as the reference genome sequence is also different from different. I know India have gone for the 10,000 genome project as well. In Sri Lanka, we are like something very down the line. It's like only one reference sequence. So first of all, we have to take like into consideration. First, we have to do the scientific background, what we can do. Then we have to emphasize that for the clinician, first of all, because most of the clinicians still, they are not in a position to go for the genetic studies. So we have to make the awareness, first of all, and then plus we should have the grants and everything for that.
Thank you. Any other?
Yeah, I mean, it's a very complex issue. The first time the concept of personalized medicine started several years ago, several decades rather. I started with the use of anti-tubercular drug, isoniazid. Isoniazid was the antibiotic which was used for tuberculosis. 45% of US population was fast acetylator because isoniazid is detoxified by N-acetyl transferase, whose level was much more in some individuals than others. And particularly US population they studied way back decades ago. And hence they said that if you have to determine the dose of isoniazid, it has to be individually specific. And that started at that time, but it's very difficult to make a drug for one person. No company is ready to do that. The only thing that for research one can do it, one can make an average of, I mean, if you prescribe 500 mg paracetamol to young adult and old, now you have geriatric medicine. You have to have a dose for geriatric patients separately. So one has to be very clear, clear on using sometimes these terminologies. It's very difficult issue. So in India particularly, or even the world, not much has been done as far as actual practice of using personalized medicine. Thank you.
Thank you so much for giving me the opportunity.
Thank you, ma'am. Moving ahead, we have our third lecture in the series by Dr. Rajiv Singh sir, a scientist, Regional Medicine Research Center, ICMR, Gorakhpur. The topic for his presentation today is RT-RPA, that is CRISPR, based one-pot molecular diagnostic assay for detection of chikungunya virus. Please give a warm welcome to Professor Dr. Rajiv Singh sir.
Thank you. And thank you very much to the organizers, especially Dr. Sunil for inviting me for this talk. So, as Madam has told, I am going to present my work where I have developed one kit which is based on isothermal amplification and CRISPR-based detection technology for chikungunya. So before coming to this topic, I think everybody is aware about this point of care testing in students, yeah, or not. Basically what we are seeing that if you are going to the clinician, he is recommending some test. So you are going and giving your samples to the sample collection center, and then that sample is going to any well-equipped centralized laboratory. And then their sample processing taking place, analysis, report making, and then this result is delivered to the clinician or patient. So it took time, mostly 24 hours, 12 to 24 hours, and sometimes more. But if we want to treat patient timely, so there are tests coming which are called point of care testing. So meaning of this point of care testing is that it should be near or at the site of patient care. So such kind of tests are called point of care tests. So now trends are increasing for point of care testing. And I think everybody is seen somehow one or two tests in nearby, like in diabetes patients. Now they can do their testing of glucose by pricking and one drop blood, and then there is chip, and that chip is inserted in the device, and that is giving the result. And one common test is pregnancy testing. Most of the people can do by dropping 2-3 drops of the urine, and then there is pink line, or if HCG hormone is there in the urine, you can do and get the result in-house. There is no need of trained manpower like that. But still in field of infectious diseases, this point of care testing, they are developing, but still there are many challenges there. So the understanding the importance of this point of care testing, World Health Organization has given a assured criteria, ASSURED, assured criteria for which kit is fit. Which kit is fit in this criteria, those kits are called point of care, best point of care test. So for A means affordable, it should be cheaper, sensitive. If there is a small number of molecules, it can identify that. It should be specific, it should be user-friendly, it should be rapid and robust. And one important criteria is that equipment-free. There is no need of high-end equipment, and deliverable to the end user, to the patient. So if this assured criteria is fulfilling, then such kit is a very good point of care test. So like I have told you, in diabetes or some other diseases, these are very much useful. But in case of infectious diseases, there are some point of care tests available, but mostly they are identifying antibodies. If there is infection, after 1 week of infection, there is first antibody is coming, IgM. So most of the kit are recognizing IgM response. Very few kit, like in dengue, there is NS1, antigen kit is also in the form of point of care test developed. But the issue is with the sensitivity and specificity. So best kit to identify the infection in the early phase of infection, like within week of infection, is the identification of nucleic acid present. If infection is there, if any virus or bacteria or parasite is there, their nucleic acid is also there. So nucleic acid based PCR is the most important test. And in COVID era, we have seen it has proven its potential. But what is the drawback with the PCR based test? For PCR, I think everybody knows there are 3 important temperatures needed. One is denaturation, second is annealing, and third is amplification. For getting this 3 temperature, we need a sophisticated equipment. We call it thermal cycler or PCR. So this makes so there is a requirement of specific equipment and trained manpower. So this test is not suitable for the point of care testing. So in last decade, one such technology which has given the alternative of the PCR, that is called isothermal amplification. Like in our body, DNA replication is taking place at 37 degrees centigrade. And in PCR machine, we are getting 90 degrees. So like in our body system, the enzymes are like that. They can melt the DNA at 37 degrees centigrade, like helicase enzyme is there. And then they can polymerize the thing. Similarly, there are some specific polymerases researchers have discovered. One such polymerase is recombinase polymerase. So polymerase is usually used for the recombination event. When after DNA replication there is mismatch, there are some mutation is coming, then this recombination event is repairing that mismatch. So this enzyme is having one important property that is called a strand displacement property. Like DNA polymerase, it doesn't have strand displacement property, so they need a above 90-degree denaturation of the nucleic acid. But this enzyme doesn't require that. So we have used this recombinase enzyme for the development of kit. So how this enzyme is working? So this is the master mix where you can see the forward primer is there, reverse primer is there, then the recombinase enzyme is there, and there are some like ATP and other things are there. What happens when we mix this? This recombinase is making pair interaction with the primers, and then this primer is specific to any gene. It will be making pair with that gene, and after pairing, there is this strand displacement is taking place. So that hydrogen bond breaking is taking place, and this yellow one is the single strand binding protein. It is stabilizes that single strand, and then this polymerase is start polymerization of the that gene. So this is the basic concept of recombinase polymerase amplification, and the beauty of this system, it is working on at 37 to 42 degrees centigrade in only single temperature. So even simple incubator is there, it can work well. And the sensitivity, it is comparable to the PCR. You can 1 to 10 copy number of target gene is there in the sample, it can detect. And after PCR, mostly in conventional PCR, we are doing gel electrophoresis for seeing the amplified DNA. And in real-time PCR, what happens, that machine is directly seeing during the amplification process. But that real-time is much more expensive than the PCR. And in PCR, that gel electrophoresis is another extra step is there. So it is not possible in the simple laboratory. So to replace that, to find the alternative of that, so we can replace the electrophoresis or the requirement of real-time PCR. One technology which I think somebody has heard about this CRISPR-Cas system, that is clustered regulatory interspace short palindromic repeat and Cas associated protein. This is one of the technology. It was originally discovered as the adaptive immune system of bacteria against bacteriophages in 1987 by Japanese group. So what happens if any bacteriophage is coming? It is leaving their nucleic acid inside the bacteria. And then a small part of this nucleic acid is integrated in the bacterial genome. So this is making an array. These are the previous infections of the bacteriophage, and their small genome is integrated here, and it's transcribed here, making the RNA. And then this RNA is combined with the Cas enzyme. This is an endonuclease enzyme. And then if next time this bacteriophage is coming, their nucleic acid is present, and this Cas system, because it is having a small segment of this bacteriophage, it is making pair here, and then this Cas endonuclease choke that bacteriophage nucleic acid. So that bacteriophage, then bacteria can survive even after the infection of the bacteriophage. So originally it was discovered like that, but in 2012, these two scientists, Emmanuel Carpentier and Jennifer Doudna, they have used this technology for genome editing. Like in cancer, there are mutations is taking place. So they have used this technology to rectify this using this CRISPR and Cas system. And this was such a promising technology. In 2020, they have got, after the discovery of this 8 years, they have got Nobel Prize for that. And after 2 or 3 years after their genome editing effect, researchers have also used this for the diagnostic purpose. So how they use? So before coming this, we know there are 4 major CRISPR and Cas system is famous in the diagnosis or genome editing. Most famous one is the CRISPR-Cas9. It is basically used for genome editing, but in diagnostic very little used. But in diagnostic, what they use? CRISPR-Cas12, Cas13 and Cas14. If you see here, they can recognize the DNA with the help of CRISPR RNA. Then this Cas will bind here and then they will do chopping. In addition to this, they have another property which is called trans-cleavage activity. So they are specifically cleaving this DNA, but they are also having some non-specific kind of cleavage activity after binding to the specific DNA. If there is any small nucleic acid in vicinity of this interaction, they will cut it also. So this is called trans-cleavage activity, and this technology is used for the in diagnosis. So here you can see there is single stranded DNA. One side, this is the fluorescence dye, and another side is the quencher. So there is no fluorescence. As soon as they bind here, after RPA, you will find that PCR product is there. Then there is CRISPR RNA, we have a specific. Then Cas system is there. So it will cut. And then we have reporter there, which is labeled with fluorescence dye and quencher. If our specific product is amplified, then it will become active, this CRISPR-Cas system. And then it will choke this reporter also. As soon as that it will choke, that quencher will become away, and then fluorescence signal will be there. And this mechanism is we used in our diagnostic platform system. So you can see here result, like if it bind and it cut, so here green fluorescence in the presence of UV light you can see. And we also, the researchers have also developed it, you can see on the lateral flow strips, that pink line using nano gold labeled antibodies for that. So visually you can see by your naked eye. So there is no need of any real-time PCR or gel electrophoresis. You can visually. So that's why it is useful in the point of care testing. So why we have chosen chikungunya virus? Because chikungunya is very common nowadays. And if you see, it is spread by the Aedes mosquito. And if you see that Aedes mosquito is also spreading two common disease, dengue and Zika. There are 3 viruses spread by single mosquito. And mostly because dengue is very famous, very deadly. So in presence of dengue, mostly this chikungunya is underreported. Because the tests which are available in the market, most of the tests are either IgM based tests, and those tests are showing cross reactivity. So sometimes if that is a chikungunya, it is showing dengue. So that is the problem with those kind of kits. And recently what we have reported, because this is the AES prone area, acute encephalitis syndrome in the children, and we have reported here approximate 5% cases are due to the chikungunya. And we have found 25% cases are died due to the chikungunya among those 5%. So it means if there is need of sensitive kit, specific kit, which can diagnose early to the disease. So that's why we have aimed to develop this our isothermal PCR based kit, which can be applicable for the primary healthcare centers or CSCs. So this is the overall, we have developed kit. We have named this kit VRACH, that is visual 1 part RT-RPA added CRISPR-Cas12 detection of chikungunya virus. So what we did, there is what is the overall working principle of this kit? You will take the samples, extract the RNA, and then in why it is called 1 part, because in one tube, earlier what happens for amplification you are doing in one tube, and after amplification you can take that amplified product in another tube, and then that CRISPR-Cas reaction is taking place there. So that is 2-part system. But what is the disadvantage with that kind of system? Because in PCR there is carryover contamination. If you open the tube, that whole area is contaminated by that PCR product. So if there is any negative sample, that carryover is going there and it will going false positive giving. So avoid this. Now the researchers are developing 1 part kind of system. So we have developed this 1 part system where we have keep that RPA reaction in the bottom of the tube, and that 10 microliter small reaction at the cap of the tube. and we have slowly capped the tube, and we at 39 degrees centigrade for 20 minutes that RP reaction is taking place. Then a small centrifugation, only a second centrifugation we did, and then that RP, that CRISPR reaction is dropped down in the bottom, and then after 10 minutes we can see the result of either by the, and we have developed such kind of system. Either you can see fluorescence, or on that strip, chromatography strip, you can visually see pink line also. So there are dual detection system is also there. So here you can see the specificity. It's very specific system. Here we have that is the primer and probe for the E gene of the chikungunya, and we can see the specific band there. And when we did use same primer and CRISPR with the dengue serotype 1, 2, 3, or 4, there was no band. So it is very specific for the chikungunya. Here you can see the result. This is the NTC non-template control, and here green color fluorescence you can see. And as I have told, our kit is dual mode detection. Dual mode detection, as I have told, there are two kind of probes there. One is FAM biotin and one is FAM BHQ. This is the quencher, and this probe is giving fluorescence signal, and this is used for immunochromatographic result. So this you can see here. If all the components are there, then only then you can get the fluorescence, and here also this line. Okay, this is the same. And why we have used that 10 microliter CRISPR reaction on the cap? First, we keep all the reaction component of detection, that RPA and the CRISPR in the bottom of the tube, but there was no consistent result. So we have to do it in separately in one tube. So you can see that one part, 2 step is showing good result, consistent result compared to one part, 1 step. And if you see the sensitivity, this E gene was cloned in the plasmid, and you can see at least 8 copy number of E gene. It can easily detected by this move. And we have also did the extracted nucleic acid from the chikungunya virus, and 412 geptogram per microliter nucleic acid it can detect. It is equivalent to 1 copy number of E gene. So this much sensitivity. If one virus is present in your 1 per microliter, it can detect. So this is the overall our kit. It can detect 1 copy number, dual detection, 100% specific and sensitivity. So this we have published in Analytica Chemica Acta, and we have also filed the patent for this, Indian patent for this. But such kind of technology is still having some challenges. Like extraction, we are still doing with the lab method, spin column method. So now we are, even researchers are working on that to integrate, make nucleic acid extraction instrument free. So if that will be completed, then I think this will be very good technology for the future diagnostic. At the end, I will thank my collaborators, my colleagues, my lab members, and ICMR who has funded for this project. Thank you very much.
Thank you, sir. Any questions for sir?
Yes.
As you are talking about making it more friendly to the patients, so would it be possible to take any non-invasive sample? So any non-invasive sample means saliva, urine, or anything,
Sure.
and then do the lateral flow assay?
Yes.
Because
yes.
same as like pregnancy.
Like in COVID, this kit, such kind of kits developed
Because
where nasal swab and
yes, if already IgM and IgA testing is there, so I think it is possible to make far better in lateral flow assay.
Yes. Thank you. वो स्टूडेंट्स को लग रहा सब समझ में आ गया या कुछ नहीं समझ में आया? Sure.
Thank you, sir. Thank you for providing a wonderful insights about chikungunya virus. Lastly, we have with us Dr. Janesh Dubey, a senior principal scientist, Laboratory of Membrane Protein Biology, Center for Cellular and Molecular Biology, Hyderabad, India. And today, sir is presenting before us on the topic, fine-tuning functions of glutamate receptor ion channels, unraveling post-transcriptional and post-translational control. Please welcome Dr. Dubey. Thank you.
Yeah. At the outset, I'll start by thanking the organizers, the Professor Sunil Kumar and the organizing secretary, Dr. Amit Dubey. And of course, this is my first trip to this pious city, and I'm really excited to be here in this unique university where there is an integration of a lot of disciplines like Ayurvedic biology and also the latest allopathic medicines. Because as Dr. Balram Bhargava was saying, that probably the integration of both of these is going to be our USP, right? So I also believe that. So it's also a special pleasure to be here because I have people from all the three of my alma mater here. From my bachelor's, BHU, Professor Lakhotia and Professor Yamini Bhushan Tripathi is here, and probably many more. And from my master's, Devahilya University, Indore, my chair of the session, Professor Dinesh Yadav, and the last speaker, Rajiv, was also from the same place. So I'm kind of sandwiched between my junior and my senior here. And also my PhD place, All India Institute of Medical Sciences. A lot of people from AIIMS are also here in this university. So my lab, can this, there is this label, can that move away? Yeah. ठीक है. हाँ, okay. Yeah, leave it, okay. That's not a problem, yeah. So my lab works on inotropic glutamate receptors for last 8 or 9 years, first at National Center for Cell Science, Pune, and now at CCMB, Hyderabad. And my apologies to the senior people in the audience. I have tuned my talk to the students. So if some of things you find basics, I apologize for that. So what are glutamate receptor ion channels? So we'll go into that. I'll give you some brief introduction about that. But before that, I want to start by showing you this picture of CCMB, which was taken at probably 1987 at the time of its inauguration. So this is the institute from where I come now. So we all know that the most complex organ of our human body is probably the brain. And I think all of you would agree with me, right? And any guesses how many neurons we have on an estimate in our brain? Around, yeah, that's there. So around 100 billion neurons, okay? And these 100 billion neurons make more than quadrillion of synapses, right? So these neurons connect with each other. And there is an information transfer that happens between one neuron to another neuron, right? And around 90% of all the neurons in the brain, or all the synapses in the brain, are of glutamatergic type, right? And when I say glutamatergic, I mean there the neurotransmitter is glutamate, and the receptors which respond to this neurotransmitter are glutamate receptors, right? So here is a schematic of a neuron and a synapse. So you see that there is this synapse from these two synapses are making a contact here, right? But this is not a physical contact. There is a discontinuity, right? There is a space in between. So And you all know that our brain communicates, or neurons communicate in form of electrical signals, right? And if you break the wire, the signal cannot go from one side to another side. So how does this happen? So this is a electron micrograph of a neuron, and here you see this is the presynaptic side where you have these green vesicles. These vesicles are packed with neurotransmitter, which will be released into this thin synaptic cleft. This is around 25 nanometer in width, approximately, right? And in this synaptic cleft itself, on the postsynaptic membrane, I think there is, just to leave it. Okay. Yeah. So in this synaptic cleft, on this postsynaptic membrane, there are the receptors which respond to the release of this neurotransmitter. And a simplified version is shown here. So this is the simplified version. You see that here is the presynaptic neuron. And when the electrical signal travels and reaches at the postsynaptic side, this electrical signal is first converted into a chemical signal, right? Chemical signal, because now, because of that electrical signal reaching the presynaptic membrane, the neurotransmitters are released. Neurotransmitters are chemicals, right? Now these chemicals are recognized by these receptors which are present on these postsynaptic side, and in response to this binding, these receptors will open, right? And when these receptors open, they allow conduction of ions. And in case of glutamate, these are cations, right? And flow of cations or flow of ions, you know, is current, right? So when you have sufficient threshold of current going through these receptors, the action potential can be now propagated in the postsynaptic neuron also, okay? So my lab specifically works on understanding how these receptors actually work at the molecular level. I am a structural biologist by training. We solve atomic model structures of all these receptors, trap them in different conformational states, different states of activity, to understand how they work. And because of their key role in neurotransmission in the brain, you can imagine that these receptors are also implicated in multitude of neuronal disorders. Name any neurodegenerative disorder, psychiatric disorders, glutamate receptors are implicated in that. They are associated with that. And so they are also very hotly pursued targets for drug development. Now, how many receptors are there? How many genes code for these receptors in vertebrates? So in vertebrates, there are around 18 genes that encode for these 4 families of receptors: NMDA, AMPER, kainate, and delta. So these 4 families of receptors form very distinct functional property receptors. The functional properties, the way these receptors conduct currents, are very different if you compare the 4 of these families, okay? Now, how does a typical glutamate receptor look like, right? So I said that I'm a structural biologist. So look at here. First, here is a topological diagram. So here you see that the polypeptide or the protein is folded into this extracellular domain called amino terminal domain, because it's at the N-terminal side of the polypeptide, followed by the ligand binding domain where the neurotransmitter goes and binds, right? And then you have the transmembrane domain, the membrane domain that forms the pore through the postsynaptic membrane. Lipid bilayer is impermeable to ions, and so this pore is needed for the ions to go through, right? So this transmembrane domain forms the pore. And now a lot of structures have been solved, and what we know from these structures is that these receptors are tetramers in nature, right? So you see there are four distinct colors, right? 4 subunits are coming together to form one receptor, okay? And what you would observe is that there is a very typical 3-layered assembly. So this is the amino terminal domain, neurotransmitter binding domain, and the pore domain, right? So this is where the neurotransmitter glutamate will come and bind. And in response to this binding, the channel will open. Unfortunately, despite such importance of these receptors in basic brain functions and their implication in diseases, even till today, there are very few FDA-approved drugs, right? And these are really very handful drugs, AMPA receptors, and there are few for NMDA receptors, and many of them are also not very specific. Some of them are just pore blockers. So why the drug development is lagging behind in this field is something that I will touch upon. And of course, there are several drugs in the target, but I'll not spend some time. So why drug development against IGLORs or these receptors is so challenging is because of the functional diversity and complexity of these glutamate receptors. So you saw that there are 18 genes and they assemble into these 4 families. Now they can form many different permutations and combinations of assembly, right? And this gives functional differences between these assemblies, okay? And on top of that, there are other variations that can happen. I will come to that. And of course, even today, we do not fully understand how these receptors work, right? So a lot of work is going on. And then any drug that you develop for these receptors has to pass through blood-brain barrier. So that is also one of the key challenge, right? And of course, there are problems like side effect and lack of disease-specific targets, etc. So I was telling you about what is the functional diversity. So of course, you could have multiple homomeric receptors, heteromeric receptors that are formed. And on top of that, I'm sure you must have heard about post-translational modifications or post-transcriptional modifications, right? So there are also a lot of post-translational modifications. There are end-linked glycosylation, there are phosphorylations, and all these can affect how the receptor works, okay? And then I'm sure you know about post-transcriptional modifications like alternative splicing. I think everybody would know, right? So there are a lot of isoforms that are generated based on alternative splicing, and each of this isoform have a different functional properties. And our lab has been trying to understand each of this aspect. And depending upon time, I'll see how much I can cover today. Okay, and then as if this was not enough, there is also a family of regulatory proteins. Lot of proteins that come and interact with these receptors when they are at the synapses, when they are traveling to the synapses, when they are trafficking to the synapses, right? And all these interactors or regulatory proteins also alter the functions of these receptors. Okay. I think I'll skip this in the interest of time. So today I will tell you a story about one of the family of receptors, glutamate delta receptor, which our lab was the first to study and solve its structure. And also, if time permits, I'll go into understanding how the post-transcriptional and post-translational modifications affect the properties. So, but before I start, for the students, I want to give you some insights into how these receptors work. Okay. So, I'll show you a brief movie about how the receptor works. So here you see the tetrameric receptor, and this is one of the subunits. And now you are looking at just the neurotransmitter binding domain. Here the neurotransmitter came and bound, and now what you observe is that this binding is leading to a conformational change. Okay, so these neurotransmitter binding domains are bilobed proteins. And the lobes are freely mobile if there is no ligand. If the ligand is bound, the lobes close to engulf the ligand, right? And of course, this closure is what ultimately leads to channel opening. Yeah. So now you saw how the ligand actually induces closure in the individual ligand binding domain. Let's move to the full-length receptor. And hopefully you will be able to appreciate how the entire receptor conformational changes are happening. So here you see the ligands have bound. And in response to ligand binding, now you see all the 4 ligand binding domains are changing conformation. And because of this, you see that there are a lot of changes happening into these linkers, which are connecting the ligand binding domain with the pore domain. Okay. Should I give my adapter? I think it is a lot of disturbance is happening. Okay. So you saw that because of the ligand binding, now you see these linkers are moving and that they actually pull the pore open. Okay. Let's move forward. So our previous work, we basically showed that there are these three stable domains or the three specific conformations of the receptor. One where the receptor is in the resting state, followed by the open state of the receptor, and then the desensitized state. Desensitized state is something where even in presence of the neurotransmitter, the receptor can close. And this is something the nature has evolved because it wants to prevent excitotoxicity to the nerves. If the receptor is all the time open, a lot of current influx can happen and it can kill the neuron. So even in the presence of the ligand, the receptor can close or desensitize. Okay. And so we do a lot of functional assays because these are ion channels. You can record currents flowing through these ion channels. And how are we doing this? We have a typical electrophysiology setup. Let me just connect my adapter. I'm sorry, just a minute because it's distracting. Okay, sorry about that. So because these are ion channels, you can record currents that these ion channels can pass through, right? How do you do that? You need electrodes, right? And these are micropipette electrodes that we use. I'll go to the last slide first. Yeah. Okay, sorry. Yeah. So you need these micro electrodes. What you do, you recombinantly overexpress these receptors in any mammalian cell. So these receptors will go to the surface, right? Now you impale these receptors with the electrode and you provide conditions which are similar to what you see in neuronal milieu, right? The inside of the neuron and the outside of the neuron, you maintain the electrolyte or you maintain the similar conditions. And now when you give the neurotransmitter, you can record the current that is going through these receptors. So for example, here you see the receptor is in the closed state. You provide ligand, which is glutamate. Here on the top, you see this line. This shows that the glutamate is being applied. The moment you apply ligand, you see that the receptor is opening, right? There is a big influx of current. And even in the presence of current, you see the receptor is now closing, okay? And what you can do, you can measure the rate of this closure, how fast the receptor is closing. And that is very characteristic property of each of the receptor subtype. And similarly, this is rate of desensitization. You can also calculate once it has desensitized, how long it takes to recover back from this desensitized state. So what one can do, you can do 2 pulses at differing time intervals, right? So here you see first pulse and second pulse, almost there is no response. Here, after some time, if you increase the interval, you see that some response has recovered. You give a third pulse after some more time, you see that even a bigger response you obtain. And similarly, as you keep increasing the time interval, you will see that gradually all the receptors are recovering, okay? And this you can fit in form of a curve, and this gives you recovery from the desensitized state. Okay. And then something else you can do is how the receptor responds to varying concentrations of the neurotransmitter, right? You can start from a small dose to large dose. We call it dose response curve, okay? And this will again give you a very typical profile. And all these properties one can use to characterize almost all the permutations and combinations of the receptors and distinguish between them, okay? And this is what we use very regularly in our lab. Now coming to the first topic, and I think I will have only time for this. So, I told you about the 4 receptor subtypes, 4 families, and this is a family called delta receptor, GLUD1 and GLUD2. It has only 2 members. And this family of receptors have been typically classified as orphan receptors because people thought that they have no functions in the brain. Because why? Because all the other receptors, NMDA, AMPA, and kainate, when you apply neurotransmitter, you see this current, right? But if you express delta receptor and apply current, you will never get anything. There is no response. So people thought that, okay, this does not work. This is non-functional channel, and probably it has only structural role, right? But as Dr. Lakhotia was saying, there is nothing junk in our natural evolution, right? There is no junk inside. Cell is too smart. It's a very expensive process to make this protein. Why would cell keep this protein and have no function for it, right? And this was the question that was bothering us. So we thought that let's work on this. Let's figure out why these receptors are non-functional. Is there a mechanistic basis by which we can address this? So of course, it's needless to say that delta receptors are also implicated in a range of neuronal phenotypes, motor functions. Their dysfunction has been linked to schizophrenia, Parkinson's, cognitive deficits, things like that, okay? So these are actually pretty important targets. Again, so the first question that you will say, all the students, maybe they don't have the apparatus to bind ligand, right? Maybe they don't have the site where neurotransmitter binds. But if you look at the topological map, these receptors also have neurotransmitter binding site, right? Although it cannot bind glutamate, but it binds other neuronal signaling molecules like D-serine and glycine. So it can bind and it can respond to those, okay? And so what we did, we solved the structures of just the neurotransmitter binding domain of these receptors and trapped them in different conformational states. And now if you compare them, you will see that these lobes can also undergo similar conformational change on neurotransmitter binding, suggesting that these neurotransmitter binding lobes in delta receptors are also functional, right? They can undergo conformational change. And if we do some thermodynamic analysis, we figured out that the strength of movement or delta G, the amount of force that they generate on ligand binding is almost similar or even more than what you see in other functional glutamate receptor subtypes like AMPA or kainate, okay? So that rules out. They have a functional channel, functional neurotransmitter binding domain. Next question would be, you would say that maybe the pore domain is blocked. Maybe the pore domain does not open, or maybe there is no functional pathway for the ions to go through, okay? And so that also one can check. This we haven't checked, but in the field people have done that. There is a mutation that you can make in this pore domain at this point, and this basically makes the receptor constitutively open. So that means it's all the time open. And in this open state, it allows only conduction of cations. So there is a proper pore, functional pore that allows movement of cations through them, okay? We also did that. What we did, we made a chimeric receptor. We swapped the neurotransmitter binding domain of delta receptor with that of another family, kainate receptor, which is totally functional, which responds to glutamate, right? And the moment we made, you see that here, these are wild type receptors, non-functional. When you make this swap of the neurotransmitter binding domain, you see that now the receptor has become functional. So this was a very definitive proof that the pore domain is totally capable of conducting ions and it can function, right? So what is the problem? Why the receptors are non-functional? And we thought that maybe if we solve the structures of these receptors, we would get some answers, okay? And how do we solve the structures? We use cryo-electron microscopy. So if there is time, I can show 5 minutes, I'll finish, okay. So there is this technique called cryo-electron microscopy that we use. And very recently in 2017, these three gentlemen, Jacques Dubochet, Joachim Frank and Richard Henderson, got Nobel Prize. And the Nobel Prize was for utilizing cryo-electron microscopy to solve high-resolution atomic structures of macromolecules in solution, right? There are other methods of determining structures. You might have heard of X-ray crystallography, NMR, but they all have their limitations. X-ray crystallography requires you to crystallize your protein molecule. NMR, there is a size restriction. You cannot solve the structures of something bigger than 50 kilodalton or 40 kilodalton. But cryo-electron microscopy removes all those barriers, right? So I will, for the benefit of students, I'll show you a very quick movie how cryo-electron microscopy works, okay? So what you do, you purify the protein of interest the way we did. We recombinantly overexpress these receptors in mammalian cells because they don't express in bacterial system. We purify them, and now you take the purified protein and make grids for imaging by cryo-electron microscopy. So let's look at this here. So let's suppose that here is your purified protein. Of course, in solution, this is a proteasome example showing proteasome. But what you will have is that you will have millions of molecules in these tubes undergoing Brownian motion, right? To image it, you need a very thin film of this protein, right? And this is the cryo-EM grid on which you apply this sample, okay? And normally the drop that you will put will be too thick. It will scatter away all the electrons. So what you need to take away excess of the liquid and prepare a very thin film of protein on the cryo-EM grid. And for that, you need these basically blotters with a fancy name called VitroBot. So you blot away excess and you plunge freeze in the liquid ethane. Now what this does, when you plunge freeze in the liquid ethane, normally if you do a slow freeze, what will happen? Ice crystals will form, right? But if you plunge freeze, ice crystals cannot form. It will all be frozen in vitreous ice, disordered water, right? And this is something that you get. And now you put that grid into a cryo-electron microscope. And now you collect imaging data. So this is a transmission electron microscope. The electron beam is going to pass through this thin sample and create image on a camera or a photographic film, okay? So let's say these are the three molecules. They will create these sort of images, okay? And one can use these 2D images to make a 3D structure. So this is the kind of micrograph that you get. You have all these molecules. You see they are in different orientations, right? What you need to do, you need to pick each of them and you need to increase signal to noise because these are very noisy, low signal. So you need to select the ones which are in same orientation and merge them together, average them together to increase signal to noise, right? So this is what is being done here. You can align them, merge them, and now you get a better signal to noise, right? And this is now only for one orientation. Let's say this orientation. You need to look at this orientation. You need to look at this orientation. You need to look at this orientation. So this is what it will do. It will combine all the orientations, and from that you will generate a 3D structure. Oh, I think that was stuck. So we solved the structures of both the family members, GLUD1 and GLUD2, is shown here. And what we observed, to our surprise, was that these receptors have a very different structure compared to other family members, AMPER, kainate, or NMDA, right? They have very different family structure. Can anybody tell what is the difference? They all look Y-shaped, right? But if you look closely, let's say all these 4 domains are ABCD, ABDC. What you observe is that how the domains are interacting at each level, okay? So look at the amino terminal domain here. You see that here AB and DC are interacting. But in the ligand binding domain here, for clarity, you look at this schematic, you see that the interacting partners are swapped. Right here, you see that AD and BC are interacting, right? So there is a swapping, domain swapping, going from one level to another level, right? And this you see is completely missing in both the delta receptor structures, right? GLUD1 and GLUD2, you see that the same partners are interacting at both the upper interface and the lower interface, okay? So what it means is something that we asked. And I think many of you bright minds would have already guessed it, right? So here you see that this structure is very intertwined, right? It's almost connected to each other. The domains are connected to each other. There is conformational restriction, right? But here what you see, these 2 arms at the top are only interacting here. And there is a chance of movement of these 2 arms, right? And this is exactly what we observed. So we could get structures of different conformational states. And this is what we observed, that there is this conformational movement that is now happening between the 2 arms, right? And what does this mean? This means that even if the neurotransmitter is there, because of this conformational flexibility, the coupling of neurotransmitter binding to channel opening is lost. Why? Because even in absence of neurotransmitter, you see that the conformations of the linkers that are connecting to the pore domain are changing, right? So it is lost. So how can you check this? We can check it by locking the interface, okay? We thought that, okay, let's lock the interface and see now if we can get currents. So that is what we did. We introduced a cysteine mutation at this interface. So if you have cysteine at this interface, and if they come in close proximity, they will form a disulfide bond, right? Now, if there is a disulfide bond, that is a covalent linkage. Now the 2 arms can no longer move, okay? And in this conformation, now if you do functional assay, you see this is the wild type, no response. But now in presence of the mutant, you see that with both glycine and D-serine, the 2 ligands, you see that the channel has become active. So this indicates that indeed the conformational flexibility was the reason, the non-domain swapped architecture, due to which the conformational flexibility was there. That was the reason why this receptor was inactive. And now over the years, we also know that at synapses, these receptors are always in complex from some proteins from the presynaptic side. You all must have heard about neurexin, right? So neurexins are addition molecules, neurexin neuroligand interactions you must have heard. So these neurexins interact with these delta receptors along with some adapter protein. And there is another group in US that followed up on our paper and showed that indeed that interaction now restricts the movement, and now these channels are also active without any cross-linking, okay? So those neurexins and cereblins are actually the bona fide binders that are needed for these receptors to be active, okay? So I think I will end at this point. So we showed that for the first time that not all glutamate receptors are built the same way. People thought that all the receptors will have domain swapping, they will have similar mechanisms of activation and desensitization. Our work showed that no, this was not the case. And we also figured out why these receptors were inactive, okay? So I will skip this part for time, but these are some of the other mechanisms that we showed. Maybe some other time we can discuss. I'll directly come to this slide because this is a conference with this theme. And of course, there is a lot of Ayurvedic formulations or Ayurvedic herbs which are known to act on these receptors. And a lot of research work is going on, but still the mechanism of action, how these are actually affecting the glutamate receptors, NMDA receptor, AMPA receptor, things like that, we still haven't been able to elucidate that. And the reason has been that what somebody was saying, that people are looking for that single molecule or single compound of this big concoction which actually works, right? And I think that will always fail in case of Ayurvedic medicine, because Ayurvedic medicine is not about that one single compound, but it's about the formulation and the synergistic effect that it has not only on one receptor, but simultaneously at multiple different receptors and pathways, right? And of course, I'm very now optimistic with Ayush Mantralaya giving a lot of funds and this government's focus on Ayurvedic biology, that now more research will happen in this direction, and hopefully we'll figure something better, like how these drugs work, right? Because China has already made a lot of investments in understanding Chinese ancient medicine, and they have actually established a lot of their medicines in the mainstream. Okay, so that's all. I would acknowledge my lab members here. The funding sources, our lab is funded by India Alliance Wellcome Trust, DBT grant. I have funding from DBT, SERB, and also CSIR. We collected a lot of electron microscopy data at NCBS and now at our own facility at CCMB. And we work on almost all the receptor subtypes. We are looking at therapeutics, we are looking at understanding how these receptors work, several different things we are looking at. Maybe some other time we'll talk about that. This is my group, current group at CCMB. And so thank you all. I hope the students were able to understand something, and if you have any questions, I'll be happy to answer. Thank you.
Okay, one question which I was not clear was that when you mutated the delta receptor, then they responded to serine and glycine. But then, so what do they do in the non-mutated form? Which is what the wild type is, what function they will have.
So sir, we mutated them to show that they have a functional domain. They have the capability to bind the ligand, and they have the capability to pass the current. And now what we have shown that in brain, they are interacting with these addition molecules, neurexins and cerebellins, which restrict the conformational movement of the two arms. And so normally they would function. They would respond to release of D-serine. So there are synapses where D-serine is also packaged with glycine and glutamate. And at those synapses, these receptors respond to it. Yeah.
From inside, you have a lot of adapter proteins.
Yes.
Besides having from outside.
The cytoplasmic side.
Yeah,
yes.
I was also curious on this locking of receptor. Is it not, say, phosphorylation or some of the modifications which might alter the conformation?
Exactly. So very rightly said. So there are a lot of post-translational modifications, phosphorylation, that has been shown to increase the conductance of some of these receptors, specifically AMPA receptors. So there is a C-terminal domain. When that gets phosphorylated, that leads to some allosteric effect on the pore domain, and that increases the conductance. But mostly these sort of like phosphorylation events are associated with the availability of the receptor at the synapse, like its turnover rate. So usually the receptor recycling, its endocytosis and then recycling back to the synapse happens in response to this phosphorylation events. And actually that is a mechanism of controlling the strength of the synapse, or what we call synaptic plasticity. Yeah. But yeah, they have role both in the strength of the synapse as well as the gating functions as well. Yeah. Any other question? Students, no question is too simple. Ask anything.
You know that I'll ask a question. Chalo. If you are not going to, are you going to ask him or should I ask you a question? You know, he mentioned a technique to see the channel behavior. Did you see those channels? Ask him what is the name of that technique. You know, it's a very famous technique, which was when it was, I mean, Jayesh, thank you so much for showing me the actual data. When technique came, it's called patch clamp technique, if I'm not mistaken.
Wholesale patch clamp.
Patch clamp technique. I think if somebody has appeared in NET exam just 2 years ago or 1 year ago, this was asked. What is the function of patch clamp technique? A, B, C, D, blah, blah, blah.
Yeah.
So you have to ask, because these are the original discoverers who are working on it. If you ask, you will get benefit, immense benefit of knowing the technology even. Patch clamp is a very good technique to study the behavior of a single channel. You clamp just the single channel and see how it behaves. Excellent technique. Thank you, Jayesh.
Yeah. Nobel Prize was given to its discoverer long time back. No other question? Okay, thank you.
Thank you, sir, for a wonderful lecture. It was full of insights, delving into the functional significance of receptor ion channels, particularly in the post-transcriptional and post-translational regulation. The findings have important implications for cellular signaling and drug development. Once again, thank you, sir. As we conclude our first technical session, moving ahead, I now request our faculty, Dr. Anupma Ojha ma'am, to kindly felicitate Professor Dr. Dinesh Yadav sir, the co-chair for the technical session 1. Thank you. In sequence, I would now request Dr. Kiran sir to kindly felicitate Dr. Ramavan Gupta sir.